Hey everyone, this is part 5 of this cell cycle unit. We are looking at meiosis today. So in the last video we did mitosis detail, and today we're looking at meiosis, and this is reductive cell division, meaning that we are changing, we are changing the number of chromosomes that are present in our daughter cells once we have finished our division. And meiosis is the generation of sex cells. So if in males this is called spermatogenesis, because we're creating sperm cells. In females this is called oogenesis, because we are producing egg cells. Now the result of males and females is a little bit different. In spermatogenesis you get 4 evenly sized sperm cells. In oogenesis you get 1 egg and then these little quasi cells called polar bodies. And polar bodies are just discarded by your body. They're not used in reproduction at all. So there's differences in what you actually get out of meiosis between males and females. And the process varies among organisms. And there's a great Prezi that I've linked below in the video description by another teacher named Mr. Kuhn. And it goes through specific haploid and diploid numbers for different organisms, as well as meiosis in just another way of interacting with it as more of a slideshow than a video. So I do encourage you to go check that out if you still have questions. But in general, right, we are taking 2 haploid cells. Remember, N represents our ploidy, how many chromosomes we have. So if I'm N, I have a single chromosome. So we have an N male sperm and an N female egg, and you merge those two, you fuse them through fertilization. So 2 haploid cells form a single diploid cell. So this would be 2N, meaning that the homologous chromosomes for the males and the females, they link up. So we have a pair for each. So if we're looking at the players, what are the players involved? Here's a cell. So this cell, its diploid number would be 6. And that's true because we've got 6 chromosomes, 1, 2, 3, 4, 5, 6, paired for the males and the females. So there's a pair here and here and here and so on. Here we've got duplicated chromosomes, right? So down on the bottom, so this is a set, right? These are a pair from the male. If you're only looking at a single like this, these are called sister chromatids. And then when we compare the male chromosomes and the female chromosomes, this pair is called a homologous chromosome. And this is what lines up in the first stage of the first metaphase in meiosis. So make sure your vocabulary is very, very clear. So we've got duplicated chromosomes. The male, right, has duplicated them there. The female has duplicated theirs, but they're not homologous. They're different sizes, different shapes. The homologous chromosomes are the same set. So this would be like chromosome number 10, let's say, for the male, and chromosome number 10 for the female. And they would pair up. So what does meiosis actually look like? So we're going to run through the stages of meiosis. In prophase 1, this is where the variation comes in. We're going to talk about this a little bit more in just a few minutes. So prophase 1, homologous chromosomes are exchanging DNA. Each gamete gets a unique strand then. So we've got the female on the right and the male on the left. So here it's kind of a blow up of what's drawn in here. And if you look really carefully, let me zoom in a little bit. We've got the female and the male homologous chromosomes are crossing over. This is called recombination. And a chunk of the male DNA, right here, right, that has now attached itself to the female sister chromatid, or the female chromatid. The same thing. A chunk of the female DNA has attached itself to the male's DNA. So the colors are swapping. And this happens on all chromosome pairs. So in humans, we have 23 chromosome pairs, and we've got crossing over happening on each one, and that exponentially increases the variation. These linkages are called chiasmata. So when you see that term, chiasmata is actually that spot where chromosomes link together right in here. So again, the main idea is that homologous chromosomes are exchanging their DNA in prophase 1, and each gamete will then get a unique strand of DNA. There is no exact copy. So if we head on to metaphase 1, their homologues, or the homologous pairs, form what are called tetrads. And it's called a tetrad because I've got 4 chromosomes lining up along the metaphase plate. Notice this is not like metaphase in mitosis, where chromosomes are individually spaced. This is pairs of those chromosomes. Homologous pairs are forming up. And then the spindle fiber attaches to the kinetochore of one of those homologues. And in anaphase 1, they are pulled backwards, just like in mitosis, except this time we're taking a pair of chromosomes, not a single sister, not a set of chromatids. And so that's why I said spindles are separating the tetrads. And now there is no DNA replication between meiosis 1 and meiosis 2. We've got, let me get rid of some of these markings here. Sorry about that. So now there is no DNA replication in between meiosis 1 and meiosis 2. So we are moving into a second prophase. And that prophase, the spindle fibers, they essentially just reform. So the chromosomes are not segregated. This is a diploid cell right now. And it's because we are not replicating between, when they line up on the metaphase plate, they line up single file, just like in mitosis. Anaphase pulls them apart, just like in mitosis. Now, instead, remember, this is happening twice because we've got 2 daughter cells that are going through the same process. In telophase 2, now we've got 4 daughter cells, 1, 2, 3, 4, that are all haploid in nature. They only have 1 chromosome from each of the pairs. And that means now that we need to fuse with other gametes. We need to fuse with an egg cell, or we need to fuse with a sperm cell to form that diploid cell. So this is really significant, and it starts to answer the question about why is there sex in the first place. This is not an efficient way to reproduce, because now instead of a single individual being able to create another single individual, I have to find a mate. So evolutionarily, right, it makes it harder for me to reproduce. But the benefit is a near infinite outcome. There is a near infinite number of outcomes possible for genetic variation. I'm going to highlight that even bolder. So we've got a number of trillions and trillions of combinations that there could be to form that new individual, and that genetic variation is an evolutionary advantage for these sexually reproducing species. So that's meiosis in a nutshell. Again, I would encourage you, let's just jump back here to where it was so we don't lose my markings. It's kind of a messy video today. I do encourage you to take a look at that video I've linked in the description, or sorry, the website I've linked in the description that talks about meiosis in detail. Go through it, add things to your notes, ask questions, and feel free to leave comments here as well.